Io direi iniziamo, prendete posto. Iniziamo che tutti arrivati a prendere posto. Io parlerò un po' in inglese e un po' in italiano, così anche per la nostra ospite e per l'uditorio in generale. Grazie a tutti di essere qui. Thank you everybody for having come today to be with us. Now I'm going to introduce the first speaker of the afternoon, and then we will get to our guest of honor. And questo è un evento organizzato dal dottorato in plasticità neurosensoriale del Dipartimento NESMOS, di cui tutti noi siamo parte. E per questo adesso darò la parola al direttore del dottorato in plasticità neurosensoriale, professor Maurizio Simmaco. Please come up here, the Chief of Sensory Neuroplasticity PhD program, Professor Maurizio Simmaco.
Just the coordinator of this I think the context of the pediatrics unit and of course Professor Thompson, because it's for us it's very important this kind of science sharing because our PhD course is a really particular PhD course because it's an integration of different areas from neurology, pediatrics, microbiology, biochemistry, and then infection disease, other surgical area. Why we decided to do this kind of PhD course? Because neuroplasticity is something affected from many different factors. And of course, in this seminar, you can see that in the evolution of the child, a lot of different factors play a role that change in intensity also depends on many particular points in order to be really recognized when the child grows, when it's out. So we believe that it's an important tool in neuroplasticity to have the contribute to the starting point, and point is detected in pediatric area. And many factors you will see will affect this kind of evolution, and probably today we will learn a lot of information. Thank you very much.
Thank you very much, Professor Simmaco, and thank you for having made it possible the meeting today. Now I will leave the place to Professor Oliviero Bruni, just Chief of Residency Program in Child Neuropsychiatry and the Chief of the Unit of Child Neuropsychiatry here at Sapienza, and Professor in Psychology and Neuropsychiatry, many time professors.
Thank you, Daniela, for giving us this opportunity to share our knowledge with Professor Thomason. And we spoke before about our, in some way, some shared interest about sleep, because the work of Professor Morrison are in some way groundbreaking, because they always go to demonstrate that something happens before the birth. And what happens before the birth is in some way conditioning the development. I mean, the work on CONNECTOME and everything are extremely important. And all the naturational milestones are related to the work of CONNECTOME and the work of Thomason. But most of these milestones are developed during sleep. So sleep is a sort of biomarker of brain maturation. So I think that if we are able to share and to collaborate, I think this would be another very important point to develop. Thank you, Professor Thomason.
Thank you, Professor Bruni. And now I will introduce Professor Pasquale Parisi. He's the Chief of the Residency Program in Pediatrics and the Chief of the Unit of Pediatrics here at the University Hospital Sant'Andrea. And he's been mentored together with Professor Bruni at the PhD program. I have to say it's very important. Okay.
I will just a few words after Oliviero and Maurizio to add. Obviously, I'm so glad to have here Moriah Thomason. And I'm very happy, certainly for all of us, but in particular for our young pediatric colleagues attending at our School of Pediatrics. And because they can have a great opportunity to learn about their intriguing and interesting research. Together with Daniela Polese, I reached her almost 3 years ago, as she's one of the most expert in the work on fetal neurodevelopment and related neurological functional findings. As you know, Daniela is studying early development in the context of her PhD program. And on the other hand, Moriah Thomason particularly has many brilliant projects in these fields. She has also an incredible equipe of very young and smart colleagues working on functional underlying fetal brain data analysis. They also study the CONNECTOME, as told before Oliviero, which is very important since it consists in a map of all brain connection. Moriah Thomason is one of several prestigious publications in this interesting field, as you can find searching in PubMed easily. I personally wrote to her to find a collection, and I was very, very enthusiastic when I knew that Daniela could meet her about 3 years.
3.5.
But fortunately, it's never too late, and now we finally have her in person here at Sapienza University. We look forward to hearing her talk, and we hope that we can collaborate together from now on. Now I will introducing the work in detail to Professor Thomason, please, Daniela.
Thank you, Ms. Brown. Okay, so we finally get to our special guest. Moriah Thomason, PhD, is the Dept Associate Professor and Vice Chair for Research in the Department of Child and Adolescent Psychiatry at New York University Grossman School of Medicine. She formerly served as Director of the Perinatal Neural Connectivity Unit within the Intraneural Perinatology Research Branch of NIH, National Institute of Health in the United States. Her published research addresses principles of neural development beginning in utero. Many of her studies address disparities experienced by monorealized individuals, and she has written multiple commentaries about the importance of population representative and culturally sensitive science. She received her undergraduate training at UC Berkeley and her graduate and postdoctoral training at Stanford and MIT in neuroscience. She is a standing member of the CPVD study section, serves as an associate editor for the Journal of Developmental Cognitive Neuroscience, and in 2019, she received the Peace Award, which is a very important award in the United States for scientists, from the Office of the President of the United States. Please come up here, Professor Thomason.
So usually sitting or standing? I feel like I can't be in the light. Is that weird if people stand or sit?
I think it's fine.
We usually stand or sit.
Whatever you prefer, we are here to listen to
Maybe
you.
I'll change halfway in case anyone's falling asleep. I'll just get up. All right. Thank you so much, everybody, for having me. And also thanks to all of you for coming and attending the talk. I hope that I keep you entertained and that you learn some good things. I'm going to give you kind of a quick showdown of like a lot of research projects really fast. And then I'm going to talk about some of the things that I think are important to do in medicine to change the way we care for people. So I'm going to go from science to predicting the future and change based on what's happening in society. So I think this group probably thinks a lot about this, but not everybody does. That so much, when you see this newborn child, so much has already happened. There's so much that has already sort of set the stage for that child's future health. And I almost think of it as like this river that's kind of entrenched and making the future happen. It's just deep over time. And so we do really appreciate that. I started by studying children, and I kept wanting to go younger and younger because I knew that the things I was looking at in those children had already started many years ago. And now in my work, I'm even going back to pregnancy because it's also a period of neuroplasticity. So when we think about neuroplasticity, there's plasticity in the brain of the mother as she prepares to be a parent, and probably also fathers too, but in a different way. So there's some recent studies looking at how the brain prepares for parenthood. So really you can just keep going back. So we started imaging fetuses, and as has been said, we've been able to conduct these studies since 2011 is when we started doing this. And the things that we're doing, the kinds of things that we're asking is sort of how do environments alter the networks before you're born? How do alterations in those networks signal that a child may be at enhanced risk for a future developmental disorder or delay? And then we can also look at cases of injury, right? We can look at situations where there's disease, prematurity, and we can ask a little bit about how that child might be at greater risk for future developmental problems. It also might signal a need for intervention. So most of the research I'm going to talk about today comes from two longitudinal research cohorts. One is based in Detroit, Michigan, and the other is in New York. And over time, we just continue to follow these kids. So these are kids that came in when their moms were pregnant. We scanned the fetal brain, and we just continue to bring the kids in and follow them. And the oldest kids are actually turning 10 now. So there's a decade of research that we've done in these cohorts. And so I just want to sort of prepare you all for what resting state fMRI is, because this is central to how we do what we do. So for many years, functional MRI was giving a task and looking for a brain area that responds. But then we sort of realized all of the brain is active all of the time. And those signals that are just spontaneously happening in the brain are very, very organized. And that structure can tell us something about how networks are connected. And the way that it works simply is here's just a random looking signal from the motor cortex. It doesn't look special. But when you do the simple mathematics of correlating that signal with all the other brain regions, so you just say what other regions have this same random pattern, it's a very organized, complete representation of the entire sensory motor system. And this person wasn't moving. It's just that you took the signal out of the motor system and you said, who else looks like this? And it's a perfect representation down to decussation and crossing at the spinal cord. Like, this is perfect. And you don't just have to, you can take this one scan, you can pull out this type of network, but you can pull out all the brain networks. So you have higher order cognitive networks, you have your primary motor and sensory systems, and you can look at how they're interacted. So this is the principle that the research is based on. And I came to this, I went to a conference it's called Human Brain Mapping. It was in Barcelona in 2011 or 2010. I walked into this room and nobody was doing fMRI in a fetus. Why weren't they doing fMRI in a fetus? Because you can't ask a fetus to stay still, right? And the fetus, no matter what I do, but I thought if we could get it to work, like if we could do it, right, somehow, this would be amazing because it would be the first chance to look at actual functional operations before birth. And so that was the insight, which has led to all this work. So today I'm going to talk about a couple of different things. I'm first going to do sort of the overview of properties, because the first thing we did was just try to understand how the brain develops. Second, I'll talk a little bit about cases of exposure and outcomes. So like I said, it's going to be kind of a fast ride, but what you're seeing here is a connectional matrix of the 10% most connected networks on the left, fetuses, and on the right, adults. And the first thing you'll notice is that there's some agreement. So even though we sort of think of this as a very immature brain, the total organization of these matrices has a lot of overlap, in fact, 62%, which is far higher than I would expect, in part because there's noise in these systems. I mean, these aren't the same. These aren't like matched. So the fact that they have this much overlap is remarkable. We've also discovered that there are hubs or highly connected regions in the brain. So when you think about hubs, this comes from graph theoretical analysis, a kind of network modeling strategy. These are like the main terminal, like in your metro system. They're the place where you can get through and you can connect to different places easily through these main connecting hubs. We also discovered that these hubs have increasing rich club coefficient. Again, this is just a mathematical term that refers to the fact that in this smartly organized network, those hubs tend to be preferentially more connected with each other than any other location in the network. And so you can imagine how that makes the system more efficient, right? Because if you can get from hub to hub, just like an airline flight route, your message in your brain can get from any place in the system more efficiently if there are multiple hubs and if those hubs are highly connected to each other. We also showed a medial to lateral and posterior to anterior developmental gradients in terms of cross-hemispheric connectivity. And we showed increased neural connectivity. So what you're seeing are efficiencies. So what you're seeing here is called the spring plot. And if you see on the left is the younger fetuses with the less efficient networks. And what the plot is showing you is when a single color is a subnetwork of the brain, when those dots are really close together, they're really preferentially connected with each other. And then you see this expansion where there's more crosstalk between subnetworks. This is related to another term that we use in graph theory called modularity. We see decreased modularity with advancing gestational age. So what does that mean? It means, again, that those subnetworks of the brain are beginning to talk to each other more with advancing gestational age. And this is interesting because it's actually the opposite of what we see in the transition from childhood to adolescence to adulthood. Later in life, we don't see those networks becoming more interconnected with each other. We see the opposite. They begin to separate out and become more segregated. And the interpretation of that is that they're becoming more specialized. Like if I'm a network that needs to help you with your math, maybe I don't need to help you with smelling or laughing or something else, right? That you have these sort of specialized systems, but at this point in development, you really just want to make as many connections as possible, but in an organized way. Another study that we published showed sexual dimorphism before birth. This actually surprised me by getting a lot of media coverage because there's a lot of debate about sex differences in the brain. So that was like a rapid ride through just like normative processes of development, which I just wanted to give you as a primer. And now I want to kind of go into some of the studies we've done, trying to understand different aspects of the environment or the individual child. And so prematurity is a really important topic because, first of all, it's highly prevalent. Imagine in about 10% of pregnancies. And a significant portion of these children will have some sort of a problem, developmental problem, typically mild, but some can be more severe. And the challenge here is we don't really know where it's coming from. So if a child is born preterm, you cannot take away the huge confound of being born preterm. So being born preterm is going to affect the brain, especially if you're born very preterm and you have to go through the different types of medical interventions, right? Ventilation, maybe you have some sort of lack of oxygen at some point. These things are injurious themselves. So the fact that we've studied these children and we've looked at brain differences and behavioral differences over time, we cannot totally separate those pieces. So what we did is we scanned fetuses this is just telling you what I just said. So we wanted to know if differences in the preterm brain were observable before the event of being born early. So we scanned a lot of fetuses, and then we looked at those that went on to be born preterm. And then we went back to the data and we case controlled matched based on what age they were when we scanned them, and males and females. And so the approach in this study, so I'm going to get a little bit more into the weeds with the studies I'll tell you about now. Here we're plotting the connectivity of every area to every other area, and we're looking at that as a distribution. And what we're pulling out are the areas that are most, you can almost think of as they're skewed toward being highly connected, so kind of like hubs. And we're going to do this in each of our groups. So what you're seeing is the term on top and the preterm on the bottom, and the first thing that will jump out at you is there's a whole lot more of these highly connected areas in the term-born fetuses. When you compare the groups directly, statistically, there's only one area that's significant. It's in the left hemisphere in a sort of pseudo-language area, which my neonatal colleagues on this paper were really interested in because language is one of the areas where we do see impairment. So now I wanted to know, okay, the groups are different in this important region. I wanted to know if that when you were born mattered, and if this was an area that becomes more connected with advancing gestational age. So those are the plots that you're seeing here. The answer was yes. So first of all, this is an area that gets more of this value as you go further in gestation. So this is an advancing age indicator. But also the degree to which they expressed this had to do with when they were born, such that those infants that had longer gestational periods but were still preterm, because I'm only looking in the preterm cases right now, looked more like controls in this region. So they were approaching that same pattern, which is really interesting because the area came out of a direct group comparison. The statistics that decided this area was significant didn't know how early those babies were born. So we did more in this study, but I'm really giving you, like I said, a rapid trip through studies. So one quick thing that we did was to look at, well, how is this area connected? Like what regions is it connected to? And the primary difference was really also part of that same language area. It was on that left hemisphere, sort of Broca's, Wernicke's areas. So honestly, the thing that was most important about this paper was demonstrating that differences were present before birth. It was a very small sample. We've continued to go on and do more with prematurity, but it really does highlight this idea that I'm going to keep coming back to, which is there's something very pure about being able to study the fetus. If we can get around all the technical challenges, there's something really important about taking out the postnatal confounds. So we'll come back to that. So now, all these years later, we actually have done more with looking at preterm cases, and we've been able to be a little bit more sophisticated in the approach. So here what you're seeing are developmental trajectories across a large sample. I think there's more than 100 fetuses in this analysis. And there are some that were scanned multiple times. And what you're seeing on the left, I have to keep thinking about which side it is increased connectivity. So with age. So you see age across this dimension. And what's interesting is you really do see a strong inflection at birth. And I know Daniela loves this topic right here. and it's really the first time we've ever seen this data. So this is really, really something we've never been able to image before or look at before. Because if you think about it, like anatomy does change very rapidly. Studies of plasticity in anatomy, it does remarkably change. But functional systems, all of a sudden you're getting all of this new kinds of sensory information. Your physiology is regulated in different ways. Like it's such a remarkable change for you. And the fact that we see such a steep slope here really doesn't surprise me. It doesn't surprise you, right? There's so much happening at this stage. But what's kind of interesting is it's more gradual in terms of decreased connectivity. So we did not know, we couldn't have predicted this, but the fact that you don't see a steep change here, it's a little bit more subtle, it's more of a gradual continuous line, I think is something that we'll just continue to take apart because we really don't understand this. There's so much we can do now that we've seen this picture, like go into specific systems. So Lansing, who led this work, we also really wanted to focus on the preterm population and say, is this different? And in fact it is, and it's different in an important way. So this is my interpretation of the situation. So we have our preterm cases here now in blue, positive again is on the left, negative is on the right. And what you see is that really steep slope that I told you about is really driven by the control participants, not by the preterm participants. In the preterm, it's almost like you can imagine that there is actually a steep slope prior to being born early. So this has been a debate for a long time in this community. Is the brain potentially preparing for the fact that it's going to be born early? Is the brain, because we really have only been able to measure part of these slopes. And so really this has been a debate in the field because we don't really know if the rapid development we see in the preterm brain is like a catching up or is it an acceleration? It's been very hard because we don't have this longitudinal types of data. So it'll be interesting to see if this continues to be evident and is replicated in the future. But I think it's pretty exciting to think about the fact that maybe this brain is on a completely different journey. So positive connectivity and negative connectivity doesn't look that different between our groups. So there's something maybe about making those connections. And also I should just say here, it's not totally far out to think that the brain could be preparing, because in cases of adversity, like if you look at different literatures in children and adolescents, we do actually see an accelerated picture in many different circumstances. This has been shown a lot in trauma. Children that have experienced early life adversity, like institutional rearing, will sometimes show advanced development of the limbic system, important for processing emotional information. And so there is just a basis for this idea that stress can lead to accelerated development. Possible. So stress. So this is one topic that's always been near and dear to my heart. It's critically important, and we know that stress during pregnancy is associated with all kinds of different outcomes in children. So we see differences in prevalence of neuropsychiatric disorders, altered HPA systems within these kids, and also susceptibility across multiple systems. This is not just a brain situation. It's really can be metabolic, cardiac, lots of areas of increased risk associated with stress. And what the literature has shown us is in infants, you see differences in these connectional architecture. So primarily you see differences in amygdala connectivity to areas of the prefrontal cortex. So again, I'm coming back to that same thing that I brought up with the preterm babies, which is it's very hard to tell what is part of the prenatal environment. So we talk about it as fetal programming. We talk about it as if the fetal nervous system is affected by that mom's stress during her pregnancy. But in fact, that mom didn't get unstressed when the baby was born, right? The postnatal environment is also characterized by stress, and there's differences in caregiving. And so how do we say if we scan an infant that's, say, 6 or 12 months old, that it didn't have something to do with the postnatal environment? We truly can't. And so that was another area that I was really excited to get into with this fetal work is, can we look at stress prior to the events in the postpartum? So we scanned 118 fetuses for this study, and we used a very data-driven approach where we basically allow the brain to divide itself up into different parcels based on similar patterns of connectivity. So the brain decides what its functional boundaries are, and then we perform correlations between all of those, and we let the brain basically tell us what are the subsystems or networks of the brain. So very, very data-driven approach. And again, the math is simple. All we're asking here is, does connectivity within these subnetworks or between these subnetworks differ according to mom's stress? So very simple. And the answer was yes. There were 6 different observations. 2 were within network and 4 were between networks. And I'm just going to dive into a little bit of thought about the differences we saw. So a lot of the differences we saw were in frontal parietal and striatal connectivity. This is interesting because, first of all, we know from studies of fetuses that these areas are increasing in connectivity and changing. So we know that they're relevant developmentally at this time, but they're also modified in trauma and in a number of psychiatric diseases. In fact, the frontal parietal connectivity for me and my read of the literature is actually very transdiagnostic. I don't think there's any sort of psychiatric or mental health disorder that you can look at and not see a story that resides in this network. So very interesting that this is a group at heightened risk for psychiatric problems, and we're seeing differences in utero. Another interesting finding for me here was in the frontal motor and temporal insular regions. So I do have favorite brain areas. I can't help it. I'm a neuroscientist. Like, I have favorite spots in the brain. This is one of my favorite areas. So the insular cortex is one of those hubs of total connectivity. It's important in shifting between network dominance. We're not getting into the details, but it's a really cool region. And I consider it a circuit builder. And this is a little bit far out, but it's like we were talking about at lunch with the EEG signals and the fMRI signals. This is a place where we see these spontaneous signals during neonatal development that are really important for how the brain develops and matures. It's indicative of healthy outcomes. And so the fact that it's a hub early in life, so it's central to the architecture, and that it's a circuit builder, to me says that it's very important that we're already seeing differences here that are related to maternal stress. So I had this thought, right? I was literally on a train. I remember the moment that this happened. I had this thought. We also know that babies that stress during pregnancy is associated with preterm delivery. And I wondered if it was possible that the degree to which there was a stress effect. So bear with me. This is kind of a little bit complicated, but once you get it, it makes sense. So there was a degree to which we observed a stress effect in each of these individual fetuses, but they all varied. Like you could be a fetus that had this effect really big, or you could be a fetus that had it really strong. It's not a perfect correlation with stress. So the degree to which I want to say that it became biologically embedded, it's a little far out, but like really that you had that phenotype of stress, was it related to how early you were born? And it turned out that it did. That's wild, right? The degree to which you had this stress picture in your brain, the magnitude of the effect within you was related to when you were born. And you can see that it differs across some of the networks. So we could actually begin thinking about where in the networks was this embedding related to when you were born. So it's kind of far out there, but I like it because it's just a different way to look at data. And I'm not showing it, but in this study, we also looked at health factors and other things to just see if there was something about that where we could think about intervention, right? Because it's not easy to tell someone to not be stressed, right? But there might be some other things that were related to the degree to which the stress embedded. And we went into that in the paper. So this is another postdoctoral fellow in the lab. She's actually starting a faculty position this month, so very excited for her. She, in her postdoctoral work or in her graduate work, looked at babies that were born to stressed mothers and looked specifically within a frontal amygdala circuit, part of a limbic circuit that's important for emotion processing. And she found differences in ventral medial to amygdala and dorsal anterior cingulate to amygdala connectivity. And we wondered if the experiences, I'm sorry, I said that wrong. It wasn't stress during pregnancy that she looked at. She looked at intergenerational stress. So was the stress of a mother while she was a child, so maltreatment when you're a child, was that related to brain development of your child? So this is intergenerational risk transmission, and this topic is really growing. So now that I've said it to you, if you haven't heard of this field, this is a growth area in the field, but it's this idea that things can be handed down over generations. We don't totally understand how or why, but it's going to continue to become clearer. So this was a collaboration with Marianne Vanden Heuvel, former postdoc in the lab, and Catherine Monk, phenomenal researcher at Columbia. We, in the Detroit sample, had moms fill out a questionnaire about their maltreatment in their childhood, and we looked at whether their fetuses had different connectivity. And Cassie led this study. And what we found was very similar to what she found in her infant study. Sometimes that slide bugs out. I don't know why. Let's see if I can get it. I'm going to skip it because I've had this. Oh, it came up. It took its time. But what's interesting is we also found that amygdala to prefrontal cortex difference. So even before birth, again, we're seeing already that transmission to the next generation. Get out of there. So a topic that's highly relevant, I think, given what we've all gone through in recent years with the pandemic, is a big question about whether babies that are born to mothers that were babies that were exposed congenitally to the virus, do they have different neuropsychiatric outcomes? So this is a topic that obviously these babies are just getting to an age where we can reliably test these things, but this is something we wanted to look at. So we recruited a pandemic cohort. And I'm just going to show you quickly what we have found when we're comparing the infants that were exposed, like their moms were infected. They didn't have COVID themselves. Vertical transmission isn't really, we know now that that doesn't really happen. But I won't get into the biology. Inflammation can be bad. And so what I'm showing you is what we found in our cohort, which was a pretty sizable cohort. We did not find differences. So if any of the lines in this forest plot are crossing the middle, it's not a significant difference between the groups. That's what you need to know. And I'm showing you a lot of different behavioral measures. These are direct measures of infant behavior. So this is not just questionnaire data, but actually recording the infants while they're doing attention tasks, or we're holding their arms at their side and they can't move them. It's emotion regulation tasks. So no difference behaviorally, which is great. We're happy to see that. There's also very little difference in all of our questionnaire data. I have a couple of stars up there so that you can see which ones are different. There's one measure that's a questionnaire measure about orienting, where they have lower orienting if they were in the exposed group. But interestingly, they have better sleep. So fewer night awakenings in the group that was congenitally exposed. That is an indicator of more mature development, just to put that in context for you. So again, we have a case where the group that was exposed is showing a more advanced sleep pattern. And then finally, we looked at the brain. And there were different networks where we saw differences in connectivity between networks, and that's shown in this connectogram as lines. And without diving into it too much, it is interesting, the patterns that we see. So one of the challenges that I haven't highlighted yet is there's not a gold standard in the fetal brain. Like, you can't put a brain in, see a difference, and say, oh, that's bad. That's too bad for that fetus, because you don't really know. It's not just a small version of an adult brain. It's fundamentally a different system, and we don't know it well enough to say this is good or bad. So when I get a result like this and I see differences between groups, I have to work to try to figure out what is the interpretation. And one of the ways that I wanted to try to get at the interpretation was, okay, well, I'm seeing differences. One thing that jumps out at me is that there is connectivity between regions that are part of a network we call the salience mode and the default mode network of the brain. I'm not going to get into the details, but what it looks like is that there's potentially a more mature pattern within those that were congenitally exposed in terms of not having as much crosstalk between these networks. So what I wanted to do is like, well, maybe we just do an analysis of these connections with age and we see if I'm correct about this. And it turned out that, I think I do that too quickly. It turns out that, so here the COVID exposed are on this side of the plot, and you're seeing this, that they're higher here and here, the two plots on the top, and they're lower here and here on the bottom. When you look at age effects, all of them are going in that same direction, where the COVID positive, the ones that were exposed, look like they have a more mature pattern. This was only significant. This is a trend. This is the only one where it was significant. But again, it gives us some interpretation. But my total takeaways, and I haven't, that study's under review right now, that it hasn't been published yet. But my total takeaway is this was very little difference. There were a ton of tests that we did. So you saw all of those measures I looked at. If I perform multiple comparisons correction on those measures, none would be significant, because we threw everything at it on purpose, intentionally, because we really want to do a comprehensive evaluation. But my takeaway from the study is there's really very little differences at all. And those that exist almost show a little bit of a more mature pattern. I don't think we have to worry about these kids. So that's good news. So there's a bunch of studies that I won't get into, but I just wanted to let you know some of the work that we've done in case these are topics that are important to you. So these are all studies where we looked at conditions during pregnancy and how they affect the network. So we've looked at cannabis use, we've looked at toxicants, we've looked at maternal sleep. We've also looked at outcomes. So how do the networks predict the future development of the child? So we do continue to follow these kids and assess these kids. What does a pattern look like that predicts a better future outcome? And then we recently published our first paper looking at behavior during the scan as it relates to network. So the behavior we looked at was movement. So I told you that I was going to finish the talk talking about the future. So you got the data blitz of the fetus, and I'm going to transition now to sort of what are we going to do? And some of my thoughts have to do with, so we've talked about developmental building blocks, but how do we really improve health? And what are the pragmatic steps that we need to follow in order to get there? So one thing that nobody can deny is that there's been really a robust explosion in sensor technology. So all of the kind of wearables and naturalistic surveillance that we can do on our own in our homes is, oh, and this is the Gabby, this is the sleep baby, that's what that looks like. The Oura Ring, is we use both of these particular sensors. And then also we can't argue that AI is exploding, right? It's part of everything. So these two things are a lot of what I'm focusing on. So passive sensing is getting way better. Like you can use it in all ages, waterproof, temperature resistance. The Oura Ring at the bottom only has to be charged once a week. And you can measure all of these different things. And one thing I want to highlight about this is you can measure physiology, but you can also measure behavior. So things like routines. Does this person go to the same places? Does this person go to sleep at the same as every night? Like it's not just activity, but it's actual behaviors. And I will say we've talked to Apple because Apple has a women's health division. And then think of all that data, right? Because that's even like your text messaging and who you're talking to. So I think we really have to use these technologies and we have to learn what we don't know. We have to learn what the patterns are. So for example, this is taken from a Oura Ring. The ring measures temperature, and I'm showing you temperature from before a woman became pregnant. You're seeing her monthly cycles here. Then conception, across her pregnancy, with characteristic hills and a pre-delivery rise. So there are signals embedded within every pregnancy just in one channel of this ring. And if you think about how high dimensional the data can be, if you look at multiple channels of data and physiology and behavior, I think you can predict when a baby will be born. I think you can predict who's going to get gestational diabetes. I think you can predict who's going to have postpartum depression. And we just don't even know yet. We don't even know. But I would be very shocked if we weren't right. So we've started doing this study, and this is an indication from our data that I'm correct. So already we can see just from the woman wearing the ring, we can predict her own ratings of stress on a day-to-day basis. So she tells us, we send her a text message and we say, how stressed are you today? And that is correlated with the signals that we're getting out of the ring. So we're going to keep doing this. This isn't funded yet, but I'm very interested in being able to do this. And I'm showing you something no one's ever seen before. The ring is related to the development of her fetus's central autonomic nervous system. So I'm very excited about this. Another thing that we're using AI for in the lab is to code infant behavior. So we can actually have infants in their home with a web camera, and you can see the little dots on the infant's eyes. And the blue dot is where the infant's gaze is looking. So we can use computer vision to code infant behaviors while somebody's at home, which is so fantastic. And in fact, this would be good in Italy because they don't even have to come in to see you. You can do this just naturally from their home. And so another project that we're working on, so it's really helped us a lot in the lab, right? Because we can really assess behavior, but this is my actual vision. The vision is that you could phenoscreen infant development and you could send reports to pediatricians. So essentially you can have an infant behave in front of a camera, you can put that in a cloud, and automatically you can send that down as a report that the family can get, that the pediatrician can get. And you know how important that is because in the United States, children, I'm going into it too much. I'm going to pull myself back. It's a very important topic because I really do think that we're going to get to this stage in the future and that the same will be true for pregnancy, that we can have this naturalistic ways of actually surveillance over health, and we can actually help care for people better. And this, in fact, is already working internationally. So that's another benefit of this, is getting into places where it's hard to do research. So I have projects in Addis Ababa, Ethiopia, and also in India, in Belgavi. So really already, because the data goes up to a cloud, you can analyze it anywhere. So anyway, this is what I think. We're really far from being able to do this because there's so much that we have to figure out. And I don't know how it is here in Italy, but in the United States, it's going to require unconventional relationships. You know, academics are over here, industry's over here. We're very separate. I think we really have to come together around these topics if we really want to change the way that we care for people. So another thing that we have to do is really big studies, collaborative studies. And so I've been talking today, this is the HBCD study that we were talking about, right? So this is an important project that will recruit more than 7,000 kids across the United States. COGEN is an international alliance that I helped to establish during the pandemic. It stands for COVID Generation. Recover is a big study of recovery after. So anyway, I can go on and on. These are all really important studies because they're longitudinal, large studies of human development. The HBCD study is doing all kinds of different things. So there'll be wearable sensor data, there'll be tons of biospecimens, brain imaging, EEG, fMRI, all kinds of MRI. And then I just want to thank all of the people that have contributed to this work. I think during the introductions, I think my team came up at some point, and I really thought, yes, it's really them. They've done so much. This is really a reflection of their work. And that's it. Thank you.
Please stay. Thank you, Moriah, for this outstanding presentation. It was really amazing, and we got, I think, incredible magnitude of information about your studies, which are so at the avant-garde also, I think. Because you have to know that when I was looking for information about early development in the passage from the fetal stage to the birth, her name was the name who came up. Solo è venuto fuori inevitabilmente as I think you are one of the most important experts in this field, because whoever I spoke about this topic, they said, you have to talk to Moriah Thomason. I went to many places, also in other states, and they said, no, you need Moriah Thomason. E tutti mi dicevano che era Moriah per forza per questa cosa. Are there any questions about whatever you think can be interesting for you? Qualsiasi cosa pensate, don't be shy, non siate timidi.
C'è qualcuno ragazzi?
Prima gli studenti.
Prego, avvicinate,
Prego.
che non abbiamo potenti mezzi. Prego,
Abbiamo una connessione internet, ci sono persone che sono connesse con noi, please come here.
Maybe it's a stupid question, but I don't understand how you consider a control. I mean, because you do usually the fetal MRI only in pathological cases, I think.
No. Yeah, here. It's a great question. It's a really good question. So a lot of our studies are typically developing typical pregnancies, but with a range, so the continuum. So like, let's say mom is depressed, right? Some of our moms are going to be subclinical, right? And some are going to be diagnosable, right? A high amount. A lot of our studies are looking at depression, for example, on a continuum. So we're not just comparing the clinical to the not clinical cases. All of the pregnancies are typical pregnancies. Now, some of our cases are more challenged. So the Detroit sample is predominantly a very low resource and very stressed, highly trauma, really challenged community. So just recruiting a healthy pregnancy off the street is different. Like, it's not a typical United States representative. So does that kind of get at it? They're not patients.
Okay.
Yeah.
As you said about the COVID infection during pregnancy, affecting particular the frontal region, do you have any data about an increase of ADHD in next year?
So I am one of several labs. Even Livio Provenzi is in Pavia and has done a lot with COVID versus not. And in fact, he sent me a WhatsApp message today, coincidentally. I was like, I'm in Italy. So But he has a beautiful cohort where he's been looking at outcomes, also gene expression, so different people looking at different things. And I think that the picture is there's not going to be a signal, that we're not going to see differences, that these children are going to not have concerns. There have been other viruses in pregnancy that have had a huge immune response, and those children have been devastated. I really don't think that the story with COVID is going to be a problem, because it's not just my lab that's finding this, it's the other labs that are beginning to have the follow-up. Now we need long follow-up, and there are some epidemiological studies where they looked at large medical record systems. There's a study out of Brigham, or at Boston Children's Hospital, essentially, with like thousands of records, and they did see a signal of risk, enhanced brain injury types of risks. But really, I don't know if it's going to continue to be the case, because that investigator even concluded that the confounds were really working against the study. So, yeah.
Vaccination and then the
Yeah.
disease.
I
Soft
really,
disease. It's so confused, the picture that you cannot have.
I really scoured the literature recently, because I was presenting this at a conference, and I really tried to give a summary, and there's just not a compelling case that there's going to be a concern, which is great. I think that's what we all hoped, but.
Thank you very much, Professor Thompson. It's very impressive to see, to touch how fast go the science and the possibility. This the last part about eyes recording and the possibility of would change completely the pediatric evaluation. But this is when you talk about this intergeneration passage of the this means that we can have such epigenetic modification like in Holland in the Second World War.
Yeah.
So the same mechanism, the same effect.
That's one of the most suspected, pathways. So a lot of folks are interpreting it as epigenetic. Yeah, that's the most studied. How did this happen? Right? That's like the most studied.
It's complicated, of course, to study this kind of process because we don't have the mirror gene, or at least something to follow.
Yeah.
It looks like the same mechanism.
And I think it's because the epigenetic changes are going to cause different developmental changes, right? So like a tryptophan pathway, or right, because it's going to change the way things are expressed, which is going to change how things are built.
You said the tryptophan pathway.
I just made that up though. Like don't attach too much to it.
No, okay, because
but it's important in development.
Absolutely.
Exactly.
Okay, but because this pathway is supposed to follow and impact a lot in the. Okay,
That's why I like the example, because it's a pathway that would impact development, and then the gene expression could be passed on. Like it could just be that's.
Yeah, the recent paper in Nature shows the correspondence between IDO and.
Between what?
The IDO, the endogenetic oxidase. That changes the expression and affect the.
And it interacts with microbiota. Is that?
More or less, but also in the brain function.
Yeah.
Thank you.
Yeah. I think you can reach it.
Thank you very much for the lesson. It was very interesting. I would like to ask you a question. You talked about modification, the brain connectivity through the years. But I'm interested to know what happened in the few moments during the birth of the child, and if you think, what do you think the trigger factor that guides this transition could be?
That's
So bizarre.
No, it's a terrific question. So first of all, that hasn't, I think probably if I was trying to study at the moment of birth, I probably would use something like EEG. I'd probably use a different method, because a scanner is a big clunky thing, and it's much nicer for the family, I think. So don't like take my new baby and put it in this, right? Like you don't have your baby. So I think probably EEG also because of the high temporal resolution, right? You don't get that with the slow signals and you're measuring much of hemodynamic. So that's another reason to look at EEG. Like this is interpretation of oxygen delivery, which is a lot different than electrical. Yeah. I think that's the way I would do that study. And what was your science question that I can't answer? What was that?
The second one.
Yeah.
Yeah, if you think that, I would like to know if you think that some, what you think that trigger factors could,
No, it's a beautiful question because I don't think we know, but I think it is sort of this magical kind of in our reach, out of our reach question in science. Because there are things that happen at birth, like people talk about cesarean versus birth. And they talk about cortisol being different in the two types. We know that there's certain neurotransmitters that change at this transition. So GABA and glutamate change in terms of being inhibitory or excitatory. Why? So I think there's magic there. I don't think we totally know. And I don't think that, I'm not aware, and maybe somebody is, of what signals that. Like what's the cause that causes that neurotransmitter to change? I haven't looked at this area of literature in many years. It's like going back to my graduate training, but I've always been fascinated. Do you have an idea? This is her passion. So, what do you think the trigger is? Does it happen before you're born? After you're born?
What I can say is that, in science, generally we have been focused very much on the fetal organism and newborn, while there are just a few articles about the incredible impact of the extrauterine environment on brain, which is pretty simple, but it's not very often considered, because we always focus on what happens inside the brain without considering the relationship between the brain and the environment around. Maybe we consider even more the womb, the utero environment, while we absolutely, it's like you're blind on the extrauterine environment, which is full of stimuli. Somebody is called stimuli bombardment. And the brain, the neural cells are the more reactive and sensitive to external stimulation. So this interaction between the brain and external environment make this that you have just called in your brand article transformational step. You have mentioned the transformation, which is a very specific word. It's not philosophy, very science. It means that there is another form, another existence which was not existent before. I can just, say that I am also a psychotherapist, and I have worked a lot with this human birth theory by Massimo Fagioli, and he wrote, by deductive method, he stated that there was a stimuli bombardment in the external environment, but there is only one stimulus which really can get immediately the brain, justifying the sudden surge that you described as well in your article, and which is not matter, but which is energy, and it is light. Light can get the retina, and the circuits, the retinal cortical circuits are already fine, and they are fine, they are complete exactly at the same time when the fetus is viable. So there it is. Yeah. So it can be a very, very brilliant hypothesis, but we should demonstrate it.
Yeah.
I don't completely agree.
Yeah, perfect.
I think that the light is crucial, can be important, can be crucial. But don't you think that we can have a mental activity, autonomous mental activity during the last months of the pregnancy? According to your functional study.
I love it. So it is interesting. When I first started writing these papers, I was like one of the only people doing this work. And I would sometimes have reviewers, because you have reviewers that aren't doing the work, right? And I would have them look at the areas where I had a difference, and they would really, the word I use is anthropomorphize. So I don't know if that word translates.
Anthropomorphize.
Yeah. They anthropomorphize the brain. They know what they think the brain is doing, but it's very like from your own perspective, right? And it's from what an adult brain does in that region. Like a language region, for example. If I see a difference in a language region, is that language region thinking about language or trying to process language or having the same kind of mental states? No, because what a brain needs to do when you're a fetus is develop. Like it needs to grow. It's not concerned with the same types of shopping lists or episodic memory or the things that we're thinking about. And it's not the job that it needs to do. So I tend to think like, if I was designing, like as a designer of a system, which is part of why it's interesting to study the fetus, like how do we start? What are the building blocks? I tend to think that little bit of tissue just has a really different job to do than you or I think of when we think of these regions serving functions later, like cognitive functions, things we think about. And in terms of there being a point at which those mental states begin to approximate the kind of things that you and I think about when we think what a mental state is, we're really pushing our experiences back, right? Like we're thinking what mental state means based on what we understand of experiencing it, right? And so what does it, I don't know if that means the same thing as what kind of cognitions a fetus might have or what kind of things it might experience, right? Like at some point we don't know the definition because, yeah, I hope I've explained it. Like I think that they can't have the kind of mental states we can have, but should we not think of those states as having some sort of mental state meaning? I don't know. Yeah, it's complicated. I like the discussion though. I like that there's some disagreement. Like this is exactly what should happen in a classroom, so this is good. And questions we can't answer.
Yes.
I have a different question, but I mean, do you think in the future we will be able to use your studies, I mean, the MRI scan to predict the psychomotor development of the child, and maybe if you would develop some like pathological development like autism or ADHD or something like that?
We are doing those studies now. So we have like kids that are somewhere on those spectrum, right? Again, we're not recruiting them on the basis of that, but I do have a postdoc focus on autism right now. But the answer is no. Like I've never thought that we're going to put kids in a scanner to diagnose them. Like it's such an inaccessible technology. So that's why I kind of like move in my career and in my thought process toward the end of my talk where I talk about what are we going to do. I think you use these systems to understand the unexpected or to answer some of these types of questions. And when I see that, the way that I think that they'll help us is like, what if autism is represented? So we know autism will be represented somewhere in these networks. There's some way in which we'll do the right different kind of experiments, us and others will like map out how much we can possibly know about the earliest signals of biomarkers of autism. We're never going to run kids through a scanner to then detect autism. But what might happen is we might see something unexpected, like that there are certain regions or circuits that are involved that we just wouldn't have thought of because they don't relate. Like a lot of the findings in I'm going to say something very depressing, so I hope you all follow this. Like a lot of our findings are just self-congratulatory. Like you just look at the system that you think should be affected and you're like, oh, like, you know, you have depression, the systems that process emotion are involved. Like, okay, great. Like it was what I expected. Or like in a autism brain, you see maybe the things that represent the social world, right? Or stimuli. Those are the areas where people go write the papers about. Because it's what I thought. Like the kinds of things that are implicated in this disease are the same circuits where I see a difference. Like it's very, that's why I say self-congratulatory, because we're confirming things that we expected to be true, and it's a little bit biased. But what about the things that we didn't write about? What about the stuff we didn't highlight when we wrote the manuscripts? And I think that's where it could be really interesting, because it could be that there are signatures or fingerprints in diseases that will tune us into something else that actually could become a target for intervention that isn't the behavioral manifestation of the disease. Imagine. Or maybe a gene therapy, or maybe like, I think it's what we learn about the representation of disease in the brain, and that we may be able to treat things differently that behavior would not. That's my interpretation. And when I write or read people's papers, I'm thinking that way. It's like, how do we use these fancy machines?
Great, thank you very much. That's so cool. No, Alessandro Veretti.
Thank you very much for your brilliant talk with a big number of insights to explore. My kind of interest is genetic disease and the degenerative disease, genetic one. And I'm curious to understand how all this discovery can interact with genetic basis. And in particular, could epigenetics enhance these discoveries? Because, of course, the environment is important, of course, but genetic basis too. And the genetic panel of next generation panel for neurodevelopmental delay is one of the biggest that we have. So how these two fields can match together?
It's really challenging, usually because of sample size, and you study rare genetic disorders, so you know that well. There have been some initiatives that I think can move us toward, but these are very high dimensional data already. I didn't even get into like the omics aspects of the wearables, even very high dimensional. We're talking like hundreds of thousands of time points. So when you analyze behavior, it's not different. Like, we're doing some projects where we're trying to use computer vision to like watch videos of children. We'll have like 7,000 hours of data, right? So when you try to say, can you put imaging and genetics into the same space, it's really complicated without huge numbers. Now, to our advantage and the future of where we're all going, there are initiatives like Enigma, which started, I don't know, it's been like 15 to 20 years ago, which is, Enigma started with genetics and imaging. It's now many other things, but it was basically people putting data into a common place from all different studies that had also genetics. And so now it's become this massive resource. So I think we can get to a point in science where we can begin, but we really have to have huge data and clean data, which is really hard. Like, the more data you have, the task of keeping it clean and quality controlled gets bigger and bigger. Do you find that also in genetics? Like, the bigger the data, the more you need to just do validation and quality control, and all these pieces get really important. Yeah, but I think that's kind of a limited answer is the size. I don't know how far we can get.
A very basic question. How do you manage the movement of these babies during the functional MRI, which will last at least 4 or 5 minutes for each position, I guess?
Yeah.
So how many dropouts you have? How do you manage? Because I think the organization is very difficult.
Thank you. You sent a slide about how hard it is to do the studies. Because it's not just, I'm going to talk about movement to answer your question, but I'm going to highlight the other things. So some of the other issues you face it's really small. Like the actual resolution, right? The brain is really small. So the resolution of your camera taking a picture of something further or less far away, like you're averaging across different spaces. And you can't keep making your voxels smaller. Voxels are three-dimensional pixels because the voxel has to average over a space of tissue. It only goes so far. So you have a resolution issue. You have the fact that mom is also moving because she has respiration. You can't ask her to not breathe for 7 minutes or whatever. So you've got her motion. You've got the fetus's motion. You also have an imperfect understanding of the hemodynamic system. So neurovascular coupling is the basis of our signal. It's an immature brain, but it's the neurovascular properties also not totally worked out. So we have a ton of issues, right? The answer is the fetus does alternate between periods of quiescence and periods of motion, and it's about 50%. So with all of the cleaning that we do and all of the kinds of steps that we do, I was talking to Daniela, was saying, why did it take you so long to publish the study that you wanted to do? And I told her it's because we do experiments on our choices during processing. So we'll use different thresholds, we'll look at what gets dropped, we'll try different things, we'll make distributions, and then after a lot of sort of testing our own method, then we say, okay, this is the method. And we haven't published it yet, but all of the things I do are shared publicly. So even the code for the infant vision, that's all available publicly with like how to do it, so other labs can do it. I will say one of the coolest advances that we've made in my lab in terms of dealing with the motion issue is you have to segment the brain. So I take at least 12 minutes of data to get like say 6 minutes, but I actually take like 24 to get like closer to 12, because again, it's just, you better quality, you get more. But one of the things that we did is you have to find the brain, and the computer programs that you process this fMRI data with, they can't find a fetal brain because it's encased within this whole other beautiful picture of mom, and the programs can't pull the brain out. So that was a huge problem for us because the fetus is moving. So we couldn't even apply a single mask because at every time point, you had to take a 3D structure out, and solving that problem was really complicated. But we were able to use deep learning and convolutional neural nets, and we were able to train our system to find the brain. And then like a cookie cutter, we could get the motion, we could make a perfect mask, and we actually could apply motion transformation to a perfect mask and pull the brain out as a complete. And that was probably like the greatest advance in terms of our ability to process the data. It's really hard to process. It was taking us like 30 hours per case. So, yeah.
It's very important because it's totally new to us how to scan fetuses.
Getting the data is easy. They're very compliant because they're going in the scanner. So collecting the data is very easy, like much easier than anything else. Right.
No? Any other questions? No? Okay.
Thank you very much. Thank you very much. Scientific sharing of the knowledge. So thank you very much again. We hope that we can send some PhD student to you. Thanks.
Your residents. Thank you for your generosity and thank you very much again. Thank you, Moriah.
Yeah, I admit it.
Okay, perfect. Yeah.
Se mi pedivate la storia lunga.
Some of these students are using these sheets as not so they also have yes. Wow.
A livello neurale per la nascita degli altri.
Come stai nascendo?
È una cosa più terra-terra. Per caso c'è il team qua?
E loro iniziano sempre con queste cose.
No, una pazza magnifica, quindi sappiamo un po' come è andata.
Sì, perché magari potrebbe a un certo punto provare
Facciamo...
sulle classi. Forse va bene, che recentemente gli abbiamo rubato 7 femminili, quindi stanno in classe.
Siamo al sistema con
Very engaging. Ho detto, premi quando vuoi. No,